This is why in emails I've said to people in the past, I'm not doing Wolbachia stuff anymore. I'm not working with Wolbachia anymore. Good morning, good morning, good morning. Hello, hello, hello. I am John Beckman, Professor John Beckman, Dr. John Beckman, the father of Sid, the transgenic mother of sin, your hostess with the mostest. Get a green screen in the back. No coffee today. It's not black. Have my brain tab behind my back just to make it fair. Normally, I do not react to preprints, but I'm really interested in this research. It's a fascinating new field, and I really want to talk about it, and it fits really well with both the class that I'm teaching right now, and it also fits really well with the content that my viewers like to see. It's got bugs, it's got engineering, it's got creativity, it's got everything you need to be cool science. So the paper I want to talk about today is, it's probably getting submitted to a higher tier journal. Some of the authors are very, very famous. Takema Fukatsu, very famous in entomology and symbiont biology, very famous, world-renowned researcher and friend, and first author and co-corresponding author, Ryuichi Koga. Many other authors, a big collaborative work on this paper. And the title is A Single Mutation Makes E. coli an Insect Mutualist. So let me first try to discuss some of the context in which this paper pops onto the field. So many symbionts are extremely difficult to work with. And the reason is because when you have symbiosis, you've got an organism A living in very close proximity to an organism B. And usually the symbiosis involves a process where one is giving one to the other, whatever the organism is. And sometimes what can happen in symbiosis is just like what happens in capitalism, is specialization, where A gets particularly good at doing one thing and B gets particularly good at doing a different thing. So then A just says, well, why don't, let's say A makes X, and let's say B makes Y, some random variable, something, some metabolite, some nutrient source, some vitamin, some amino acid. Let's say A is really good at making X, B is really good at making Y. And so B says, well, I'm just going to give you Y. And A says, oh, that's good, I'll give you X. This is how symbiosis starts happening. The other side of symbiosis is that eventually A says, I'm going to stop making Y. I will stop making Y. And B says, I'm going to stop making X. Okay. Now, what happens when this decision is made in A and B? What this means is that if you split them apart, now A dies and B dies because they were receiving nutrients from each other and they stopped producing them by themselves because it was more efficient, more energy efficient for them to work together. So when you split them apart, now they die. Okay. That's an obligate mutualism. So if you zoom in at the genomic level, the genomes, what's happening to the genomes? Symbiosis often selects for genome reduction. You're throwing out genes that you're getting from the other place. You're just throwing them out. And if you're a bacteria, you might do this because you want to replicate faster. You don't want to consume energy making a product when you can just take it from your environment or your cohabiting organism. So symbiosis often selects for genome reduction. Now the problem of this is that now you probably can't culture A and B. A and B probably can't culture unless you do something like a co-culture, right? You can't culture them separately. So this makes symbionts very difficult to genetically work with. Symbionts are tough to genetically engineer because they don't culture well and because they have reduced genomes. So what are the hard lessons of Wolbachia? Hard lessons. Wolbachia is a symbiont that lives in insects. People have wanted to genetically engineer Wolbachia for a long time. And so far, that research has not succeeded. And so this is one of the hard lessons of symbiont biology, simply that engineering symbionts is extremely difficult. And so you might think of other ways that you could get around the problem. We want to get to a point, and this is where biology is going. This is where I think biology is going in the next 100 years, the future, the next 200, 1,000 years. Biology is going to entirely become about creating new organisms. At some point, we're going to have diminishing returns on what's present here on the Earth. Although, of course, like we need to explore the oceans and things like that. And of course, there's always new viruses and microbes. But at some point, just like characterizing what's out there is going to have diminishing returns. And at that point, biology is completely going to switch to engineering biology, or maybe not completely switch, but I think biology is going to tack more and more and more towards creative biology, building stuff, making stuff that doesn't exist, making stuff to solve problems. It's just going to become more and more and more like that, using the genetic code for engineering. And so one way to get around this, you're going to want situations where you have an insect and you want to do something. You want to change something about that insect, but you might not want to change the insect. And the easier way to be to, well, use a symbiont, engineer the symbiont, put the symbiont in the bug, and then have the symbiont change the bug. That's paratransgenesis, where you're changing a phenotype of an organism X, not by changing the organism, but by changing the symbiont that lives inside of the organism. And I got videos on paratransgenesis if you want to watch those. But paratransgenesis has not worked in certain symbionts because symbionts are very hard to culture, very hard to engineer. The workaround to sidestep that problem, the hack, is to take a symbiont, take a bacteria you can engineer and can grow, then make that bacteria a symbiont to replace the unworkable one. This is why in emails I've said to people in the past, I'm not doing Wolbachia stuff anymore. I'm not working with Wolbachia anymore. Like, there's no point to me, in some senses, there's no point to working with a bacteria you can't grow, you can't manipulate. Like, there's no point, unless you're going to try to solve that problem, right? But even if you were to solve that big problem, which is one of the big problems, like even if you're going to transform Wolbachia, if you're going to spend 20 years of your life to do it, I'm skeptical that it's ever going to be super efficient. I'm skeptical that it's ever going to be easy, right? You might spend 20 years of your life developing a technique that only you can do, and then you die. And then it passes and like people don't do it because it's not efficient. If there's anything I've learned in biology, like techniques that continue to get passed on are super easy and super efficient. It has to be easy, because you can't create a technology that only a genius can work with. You have to create a technology that the average person can do science on. That's the only way you're going to really succeed. So I've told people, like, I don't want to work on Wolbachia anymore because it's an unworkable bacteria. No matter how much input you put into it, like, it's not going to become, I don't think it's going to become a broad sort of paratransgenesis tool. Now, I'm not saying that the eliminate dengue things aren't going to work, or I'm not saying that those aren't vastly important. I do think those are important. What I'm saying is I don't think Wolbachia is going to be this magical paratransgenesis bullet. I think this is going to be the future. It's going to be, you have a symbiont in a bug, you just get rid of that symbiont and you replace it with one that can fill its role and also be cultured. We're not limited by what evolution gave us. We're only limited by creativity of our minds. So I love this idea. I really like this idea. I think it's a number one idea. It's a good idea. Now, what makes this possible? What makes this possible? Like, what are some of the intellectual hurdles that have been overcome that make this possible? We can create synthetic life. So this is like the work of John Glass and the Venter Institute, who have created the minimal mycoplasma cell, created designer genomes. Lots of people are redesigning the yeast genome. Lots of people are redesigning the E. coli genome. We can create synthetic life at this point. So there's no reason that we can't make an organism that does exactly what we want it to do. So this is component one of reasons why this is now possible, but was not in the past. We can create synthetic life. Two, we understand some genetic principles of symbiosis now. Now, I'm saying this vaguely. We're starting to understand these interactions more. For example, in my case with CI and CI driving or sort of being a part of a factor that's selecting for Wolbachia infections in insects, we understand mechanistically how that works. And so if you could imagine symbiosis, rather than organism A interacting with organism B, don't think about it that way. Don't think about it that way. Think about it from a selfish gene perspective. It's really an interaction of genetic components on the chromosome of A, let's say X, Y, Z, that are beneficial to genetic components on a chromosome of B and vice versa. Like, this is really what symbiosis is, in my view. I'm probably going to get in trouble with some ecologists. But I really see it as I see it at a lower level as it's not as if that whole organism A is symbiotic with that whole organism B. It's really only these subcomponents of their genomes that are interacting, that are helpful to each other. Okay. So the idea is, again, like, A is not important. Whatever A is, that's not important. What's important are the factors, the genetic loci that are contributing to this symbiosis. And once you understood that, you can take organism Z and make it a symbiont of organism B simply by taking these genes, X, Y, and Z, and putting them into a different organism. Now, all of a sudden, you're going to reconstitute symbiosis. This is going to be possible in the future. This will be possible in the future. You want a new symbiosis? Just design it on the computer, send it to a company that's going to make your artificial organism, they'll send you back a microbe. That microbe that they send you back will probably be some kind of a derivative of a re-engineered model organism, like E. coli or Bacillus subtilis, or, you know, you're thinking like a gram-negative, gram-positive, or some. It's going to be like a boilerplate chassis. It's going to be like a what you call boilerplate, which means like, yeah, these are the things you need. Chassis. And this is what I was trying to get into. I was trying to get into this research. You're going to have this setup where you're going to have these like boilerplate chassis, and you're going to say like, well, what do you want? What do you want on that thing? And you're going to say, oh, I want feature one. Feature one. I want biotin. Feature two. I want CI. Feature three. I want tryptophan biosynthesis. And then you're going to take the genes that code for feature 1, 2, and 3, and you're going to put them on your chassis. And then you're going to have a new symbiont. That's what I think the future is going to be like. And this paper is kind of like one of the first papers pushing really in that direction. And Fukatsu actually has a grant on this. Here's a web page. Fukatsu has a grant on this. The Fukatsu Evolving Symbiosis Project. They call it ERATO, Explanatory Research for Advanced. Oh, it's a ERATO grant, Explanatory Research for Advanced Technology Program. And his project is Evolving Symbiosis Project, which this is kind of the idea. Kind of. Can we, like, he says, what is the nature of symbiosis? It's a good website. Can we make it? That's what this picture is kind of indicating. What's the nature of symbiosis, and can we just synthesize it? Can you just tell me what you want, and then we'll just make it? Fascinating project. They invited me to give a seminar. That was fun. It was fun. Very prestigious speakers here. So I like the idea of this paper. I like the idea. That's the setup. So here's a bug they're working on, a stink bug. And if it's a healthy stink bug that has a good population of symbionts, the color will be a beautiful green like this. And the species is Plautia stali. These symbionts live in the guts. And the natural symbiont is a variant of Pantoia. Hopefully I'm pronouncing that right. I've talked about this symbiont before in the context of or this genus. I've talked about this genus before. There's a PNAS paper. There's a Jacobs-Lorena PNAS paper where they're engineering this symbiont. Here they're engineering that genus. I should say that genus. They're engineering that genus to express cercropin derivatives to kill Plasmodium parasites. I teach that paper. So we've seen this, if you watch my channel pretty closely, or if you've been in my classes, we've seen this genus of bacteria before. But this is a different species. And this is different because this species in the stink bug is non-culturable. The symbiont is essential for growth and survival of the insect host. Normal insects infected with the uncultivable obligatory symbiont Pantoia. So in this case, their Pantoia is not culturable. And that contributes to what I was saying before. Okay, so in this figure, here's the normal symbiont infected. You dissect out its gut, and here is the yellow bacteria that lives in this symbiont organ here. And this has a yellow color. Here's a negative control. You don't see that symbiont growing. So there's a specialized organ in the gut that's housing these yellowish Pantoia bacteria. So the first question here, question 1 that the paper addresses is, what happens if we just replace with E. coli? Question 1 is like, what happens if we just replace the symbiont with E. coli? Okay, so that's kind of like the gist of this figure 1. So here's symbiont infected, this is the E. coli infected. Obviously, there's something like missing in this one. Like the E. coli might be in there, but it's not doing the mutualism. It's not mutualistic at this point yet. Okay, here's the localization in the gut. They got this specialized organ where this yellow Pantoia agglomerans bacteria is growing. The E. coli, they colonize that region, but they're not contributing. They're not the same color, right? Then they're going to look different. But this is like, if you saw this, you'd get excited. If you saw something like this would make you excited, because when you get the Pantoia out of them and you feed them with E. coli, the E. coli are colonizing that symbiont organ. It makes you wonder, like, how does the bacteria know to go there? There must be some kind of like catching mechanism in there, something that's catching the outer membrane proteins of bacteria, because they're naturally accumulating there. So this might be like an organ that specializes in evolving symbiosis. That would be really interesting. It's this organ, it could be an organ that literally like is designed to increase the evolvability of the organism, which is really cool. Like, imagine the stink bug goes into a new situation where Pantoia is not doing it no more, and it needs a new symbiont. It could evolve by picking up different bacteria in this organism. So this is really exciting that E. coli would colonize the same spot, like the empty niche that was left after you remove the symbiont. And they write that the stink bug is smearing the eggs with the symbiont. And here's a fish seeing the symbiont on the eggs. And the symbiont is also, or the bug is also doing the same behavior with E. coli. It's also smearing the E. coli when you infect it. So again, this would also make you excited these two behavioral things would make you excited, that the stink bug is smearing it and that it's actually able to like colonize in the midgut. Because that would make you suggest, like, if you could just change the E. coli to do what it needs to do, you could create a novel symbiont based on E. coli. This is a picture describing a behavior. What they say in the legend is they say when the stink bug nymphs come out, they suck, they eat up like the outside of the egg to consume the bacteria. So how the bacteria is passed on is it gets in the gut, it'll colonize the gut, but it's not necessarily maternally passed from the gut. The way that it's maternally passed is as the mother lays the eggs, she'll coat them with this bacteria. And then as the nymphs come out, they'll eat the bacteria. So it's a behavioral vertical transmission. It's a behavioral vertical transmission. That's interesting. Again, a good system to start engineering symbionts. One of the things that you're going to notice about the best of the best, and Fukatsu is definitely one of the best of the best. One of the things you're going to start to notice in science is the best of the best. What they do is they get an idea. They get an idea and they think, God, that's an amazing idea. I have this idea. How can I implement it? And then they just start screening different systems, finding the right system for the idea, if that makes sense. And then once you find the right system under which you can implement your idea, you move everything to that system and you work on that system to exact the idea. So it's like a philosophy of not being limited by a few factors, like not being limited by your quote unquote expertise, willing to go learn new things. And also just the confidence that the diversity of biology, within the diversity of biology, if you have an idea, there's probably somewhere out there in biology where that idea is being like implemented naturally. So just having the confidence to get a good idea and then screen a bunch of systems to find the perfect system under which to test the idea. It's really nice. It's really cool to see. You see the best people do that. Okay. So question one, the answer to question one is essentially, E. coli isn't yet mutualistic, but it can colonize the gut and it can be vertically transmitted. That sets up question two. Obviously, then the next step, question two is, can we passage E. coli with the bug and with directed evolution evolve symbiosis? Now, I don't know how they're going to do this because it said it was obligate. That would be the problem you have to solve. How do they solve the problem of not having the obligate? I don't know if they immediately die. Like how many generations you get? Because that's going to be the problem. Is if you remove the symbiont and then they just immediately die, you can't do this. You can't do directed evolution. Keep reading. So to help them out, they get a delta mut-S E. coli strain. Mut-S is going to be a repair enzyme. I should just quickly check that. Yeah, it's a DNA mismatch repair enzyme. So the idea is, well, E. coli can survive. They'll just be high mutating DNA, which is good if you're doing directed evolution. You want them to mutate. So let's increase the mutagenic potential of our E. coli by using this deletion mut-S strain. So it'll have problems repairing its DNA. It'll create a lot of mutations. Hopefully those mutations will contribute in a positive fitness in some scenarios to the evolution of symbiosis. And they say, what are you going to select for? What will we select for? They say two things. Growth. One selection regime will be growth. What is it? Growth of what? I'm assuming growth of stink bug, I think. And B, the second selection regime is color. So they're selecting for green, which is, I think, the wild type proper color of healthy bug. So one question I have is, how obligate is this? It must not be so obligate that, because you wouldn't be able to do the experiment unless if they just immediately died. So there must be some like wiggle room there. It took 2 years. Here they say it took 2 years. That's the other thing you see with really, really top people. They get an idea and they hold on to that. They hold on to that idea for as long as it takes until they get it done, and then they publish. Nice. Hard to do though. So I think this is answering my question here. How obligate is this? This is answering my question. However, it took almost 2 years because the E. coli inoculated insects generally exhibited high mortality and retarded growth. So it was difficult to do. It looks like they were dying. They're getting a lot of death. Okay, so I finally found this, which is important. This is important. This is how they're doing it. They're sterilizing the newborn nymphs. And this makes sense because the way that the nymphs are taking up the symbiont through vertical transmission is by eating the outside of those eggs. So it makes sense that this can work. And this answers my question of how obligate is this? If they're sterilized, they don't get the symbiont, they're only getting 5 to 10% adult emergence rates. So it's like a 90% mortality. So that answers my question of how obligate is it? It's like 90% obligate. And without that 10% survival rate, they would not be able to do this experiment. Like, you couldn't do it if it was 100% death. So this is what allows this little sort of like, well, it's 90% obligate. This is what allows them to do the experiment. So again, it's actually like a beautiful experimental system because it has this feature where you get rid of them and 10% will still survive. If it was 100%, you couldn't do the experiment. This is good in that it's a validation of sort of, again, if you have this of the need for this symbiont. You wouldn't expect this to be easy, which is what I was skeptical of. You would not expect this to be easy. So it's good. It's good that it was difficult, because that suggests that when you remove the symbiont, they really start to have problems. But it's bad because it was hard to do. But nothing that's worth doing is easy. So, okay, here's figure 2. Okay, they're testing a few different things here. One, they're testing selection regimes, conditions. And by that, I mean specifically color versus growth. So like speed of the growth or how well they grow, the emergence rate. That's what they're testing for growth. So that answers that question. The second thing that they're testing is they're testing whether the delta mut-S is better or worse. So they have a delta mut-S versus their negative control is delta int-S, which it's a phage gene, integration gene, should theoretically not do anything. So this is like the negative control. And they're testing, do they get better evolution with the increased mutagenesis? Which I'm assuming they do, because I'm assuming that's what I'm assuming these red things in the mut-S, I'm assuming those are ones that kind of like broke through, evolved through. I should read the legend though. Displayed by heat maps. So the answer to this is that it looks like the mut-S lines are the ones that are consistently producing the breakthrough symbiont evolution. So the conclusion here is that the mut-S helps if I'm understanding the data correctly. I hope I'm understanding the data correctly. I don't know how to interpret these heat maps. I see. Okay, so the key thing to understand this figure know that as you go down, you're increasing generations. So each bar in the heat map is a next generation. So what you should see in the heat map of emergence rate, if there is a breakthrough symbiont evolution, each generation should get redder. This one looks like it peaked and then got lower. I don't know what that means. But that does confirm that these are like the breakthrough evolvers. CML05 and GML07. And they're both in the mud S, not in the int S. And in this one, well, for the most part, they're like greener after a few generations. That's what I'm pulling from this figure. So again, this figure is really tough for me to interpret, but my understanding is that both selection regimes worked. This is interesting. So here's a question I can't answer. Is the color a visual ecological cue showing your mate your symbiont repertoire? That's interesting. That would be interesting. Like, is there mate preference for color choice? How do I spell this shit? There we go. Okay, so what the fuck are they talking about? The magenta shit. I don't know what that means. Edit that shit out. But here they're showing you the two that I showed you, these two that get red, CML05, GML07. I'll mark those. They get green. Here's L05, generation 7, L05, 13. It's more green. So they get green. Same with the GML07, they get green. So to answer my question was, does the green indicate health? It looks like it. So I guess the answer to which is better, selecting for color, selecting for growth, is that they both seem to work. All right, obviously now question three is, how did the E. coli change? So the next thing you do is you do genomics and you do transcriptomics. Compare versus the source stock. That's what you do. I'm sure that's what's next. Yep, this is figure 3. I can never look at these. Here's what we're looking at. Like, my issue is I never know what these bars are when I look at these figures. So, right, they're going to do transcriptomics and they're going to essentially get like a list of what genes are up and what genes are down. And those are the genes that are changing are going to be the genes that are involved in this symbiotic process. So there is an interesting paragraph. Downregulated genes contain certain transporters, sugars, maltose, ribose, trehalose, mannose, branch change amino acids. You would expect them to downregulate things that are not there, like transporters of nutrients that are not there. And then you would expect them to correspondingly upregulate transporters of nutrients that are there. They're also seeing a downregulation of extracellular matrix production proteins, which is, again, I think that's kind of expected. Like, they're going to try to evolve and live in a new environment that's totally different than the outside world. So one of the things that they're going to completely be trying to rearrange is their surface. Like, that makes sense. But it's not, again, like, it's not specific enough for me to get interested. What's going to be interesting is the convergence set, the what do you call this? The intersection, the mathematical intersection of where A intersects B. So if you have a set of the two lines that they created, CML05, changed genes, and then you have, what's the other one? GML07. Actually, the way you draw it is A intersection B. Hopefully I'm mathematically saying that right. The data set that you're really going to be interested in is this data set, because these genes are going to be the symbiosis genes. That's the data set you want to look at. I'd like to see them. It might not be possible, but it seems like they only got these 2 lines. It would be nice if they had more lines, that you could really make that way more robust. If you had more than 2 lines, then you're doing something like this, and you're like, those are the ones. And I say that with love. Like it might be really, really hard just to get the ones that they got. So I'm not complaining. Obviously it's something that a PI would say is, we'll do it more, get more data. Obviously, like that's always what people say. That's the most frustrating thing to hear as a postdoc. Do it again. So it looks like when I read, I'm not sure I'm seeing this. When I'm reading, what I'm seeing is they pulled out mutations that occurred 7 in CML05 and 9 in GML07. And one of these was adenylate cyclase, which is going to be like a huge impact signaling enzyme. I see, that's this. So these mutations are this. So if you look at their table, when they did the genomics, here's where they saw some differences. So if it's blue, it looks like this is gene frequency. So if it's blue, it's fixed. It means evolution like selected for it and it like fixed in the population. I'm assuming these are like negative controls where it didn't work. They did not see variants in these, I think. And the most consequential mutations were frame shift deletions. What is this premature stop codon? I'd like to see like just quick, give me like function. Like don't make me look all these up. Like give me a little tab here that says function. And like tell me their function. That would be nice. Because I'm gonna have to look these all up. So here's the point. From this data, they get a list of candidates. What's weird is there's no overlap. There's no overlap between GML7 and GML5. But I think that they're saying there's functional overlap between these red ones. Let me try to look and dig in the paper. So what they hoped for was the situation like this, where you get this intersection of A and B. But I don't think they did enough N to get that. You would need more N to get a higher, to get the full set. I think you need a higher N. And so they didn't have necessarily intersection of the same gene coming up twice, but they say in the writing that this and this are in the same pathway. So the other way to think about this is pathways. Instead of like actual genes, pathways changed. If that was the data, pathways changed in A versus pathways changed in B, then they have one, which is this, they say down here, which is both the Psi-A and the CRIP mutation are disruptive of the CCR pathway. I guess this is the carbon catabolite repression pathway. I don't know anything about this. So here they explain to you, what it does. What the fuck are you talking about? So here they explain to you what it does. So when there's low glucose, you turn on glucose repression pathways. And that's it. Essentially, like that causes like huge changes in global transcriptional regulation through a key transcription factor. Like you're like, hey buddy, we're not on glucose no more. And it switches to exploit other carbon sources. Okay, so then the final test is they got this overlap. The convergent data set, the overlap is this CCR pathway. So it's essentially just to become a symbiont. They're switching the nutrients that they are utilizing, which makes sense. But that doesn't answer the question of what are they giving to the bug. Like I'm still wondering that. What are they giving to the bug? I don't know. But it does make sense that this would be a first step in rapid evolution towards colonization. So then the next step, or the final step, or the next step, is mutate the CCR pathway and see if you get symbionts, what we would say, right off the bat, right away. So that's what they do. So here's figure 4. So if you see 4 figures, they're clearly going for like a high impact paper. I wish you guys luck. I hope you guys get it. So when they mutate, they make this mutation, the colonies actually look different. And I'm assuming the colonies actually, this is nice. Okay, the colonies from this single mutation, they've definitely ID'd the mutation that's causing the colonies to like change shape, change color of the ones that worked naturally. So that's nice. That's really nice. So they make these mutations, either Psi-A or the CRIP, which are in that CCP. Sorry, not Chinese Communist Party. In the CCR pathway. Those 2 mutations in that pathway are clearly changing the colony morphology. So that's the source of that. That's good. They ID'd that. And these 2 mutations right off the bat are increasing the emergence rate. And these 2 mutations right off the bat are green. So they got it. Like this is clearly, so it's the CCR pathway, which is the gateway drug, which is the gateway to symbiosis. That's interesting. Like that's cool. So the final conclusion, this is cool. The final conclusion if you're kind of like thinking about this idea, like what do you add? Obviously you add feature 4, CCR pathway deletion, which is going to make that symbiont nice. Okay, I like this. I like this paper. The Japanese groups always do good papers. They know how to do it. Let's read the discussion. We made it through. Yeah, they say it. They say what I was suggesting. They use the actual word. Evolvability. So this is the speed of evolution. Our findings shed new light on the evolvability of symbiosis. And essentially, like the new conclusion is it's way faster than we thought. Safety, final thoughts. Safety, like the idea of taking bacteria and doing the chassis idea. Like you might ask the question like, taking bacteria from a chassis and kind of like just saying, putting all these features in there. Is this safe? Is this safe? So like the idea of the synthetic symbiont, the idea of synthetic symbionts. Is that safe? Like there's a fine line between symbiosis and pathogenesis. Is that safe? I think it's exciting. And I'll say, I'm excited to see this research being done on an island. I'm excited to see that this future research is done in Japan, because Japan is an island. And so if anything bad happens, it should stay in Japan. But I definitely think this is the group to do it safely. They'll do it carefully and slowly. They'll evolve their symbionts slowly and carefully in a contained place. I'm excited. I'm excited to see where the research goes. Very good. Very good work. Good work. I like it. I like it a lot. Exciting paper.
Let the gate be open to die. I shall meet death head on. I shall meet death head on. Negate!